My name is Kenesha Atkins, and I am a student at Johns Hopkins University. I will be presenting from the research article titled Defective Base Excision Repair and the Response to DNA Damaging Agents in Triple-Negative Breast Cancer. Some background information on triple-negative breast cancer is that it does not have the common 3 targets known to fuel breast cancer, such as estrogen and progesterone hormones or the HER2 protein. This makes it extremely difficult to treat the cancer, and there's currently no effective treatment on the market. This leads to poor prognosis and death within 5 years of being diagnosed. Triple-negative breast cancer makes up 10 to 20% of all breast cancers. The objective of this article is that the researchers wanted to better understand how DNA repair machinery is affected in triple-negative breast cancer. Some background information on DNA repair pathways are there's multiple types of repair pathways that's available, but depending on how the DNA was damaged depends on the pathway that is used within the damaged DNA site. In this particular paper, the researchers focused on the BER, which is known as the base excision repair pathway, and specifically they honed in on the protein involved, which is the XRCC1 protein that is used within this pathway. The researcher team hypothesized that an indication of base excision repair defect can be seen in the changes of expression levels of protein XRCC1, which could be targeted to provide new treatment strategies for this aggressive breast cancer subtype. One of the questions that the researchers wanted to have was if this particular protein is expressed in triple-negative breast cancer. So they used a database called the Cancer Genome Atlas, TCGA, which already had this information, and they specifically looked at the expression levels that this protein had in triple-negative breast cancer. And they concluded that there is an increased production of this protein in triple-negative breast cancer, which was unique compared to non-cancer cells. Next, the researchers wanted to look at the base excision repair proteins present in the cell lines that they picked out to look at. And the 3 out of the 4 cell lines did indeed show increased levels of the protein XRCC1. So this confirmed the already set data that showed that there is an increase in the expression levels. And when they performed this experiments themselves, it correlated or it aligned with the data that was already present. Next, they wanted to look at the ability of the DNA to repair itself through this base excision repair pathway. So they looked at the repair using a specific experiment, the fluorescence multiplex whole cell reactivation repair, and they concluded that most of the cell lines with the triple-negative breast cancer cells had lower BER repair compared to normal cells. So based on the experiments performed and thorough analysis of the results, it was concluded that the expression level of XRCC1 alone cannot determine the extent of DNA repair defects. I think the research was executed and interpreted well. It was very easy to follow their thoughts process, and the techniques they used to answer their questions was well thought out. I also appreciated the reflection they provided as far as not looking at DNA repair defects in a one-dimensional way, but rather to understand that multiple proteins and other factors or pathways are all potential contributors to triple-negative breast cancer. If I were to continue this research, I would move this into an animal model to see if I would exhibit the same results that was seen in the cell lines. It is very important if you want to have a treatment go to clinical trials that you have an animal model to prove some of the data that was seen in the cell lines. Thank you.